Good morning or evening or afternoon, I guess, wherever you are. No clue where you guys are. I mean, you guys are MCAT sickos. You're probably up at 3 in the morning studying or watching this. But my name is Nate. As I introduce in all of my videos, I have a website that I've been creating and releasing a whole bunch of free resources for the MCAT, whether it just be basic tips, but also a whole bunch of videos that I'm working on, a whole bunch of my notes, a whole bunch of cheat sheets, things of that sort. You can find the link to all of that stuff in the description. I also do offer tutoring on my website as well if you are interested in that. But the whole goal of these videos is to provide a specific explanation of all of the content that you need to know specific to the MCAT. I'm hoping for this to be a pretty comprehensive review of all of the information, explaining every single thing that you might need to know so that you don't necessarily have to keep coming back to the video. So because of that, my explanations are going to be a little bit more in depth. These videos are not necessarily meant to be a quick and short little introduction. They're supposed to explain everything you need to know. Additionally, because of that, I do recommend watching this on a computer or some sort of TV. Like if you just have like a 3,000-inch flat screen TV floating around, it's probably a good idea to throw this up there. There's a lot of text and a lot of talking that I do on my slides just because they are meant to be comprehensive, a little bit more of a lecture style rather than a just quick little animation or something like that. And so just by watching it on a larger screen is probably going to help out a bit with some of the smaller text on my slides. You can also, of course, play around with the video speed in case you want me to be talking faster or slower, whatever works for you. I do also have a link in my bio that will take you straight to a page on my website that I have notes that support all of the videos that I make for the MCAT. All of my metabolism videos in the future, I'm probably going to be making some videos about physics and random things like that. I do offer notes that pretty much list all of the information straight in my slides, but just already pre-written out on a page for you. You can also take my graphics, use whatever you want for those. They are going to be watermarked just so no one can try to sell them. They're free. They're meant to be free. That's why you'll see a whole bunch of my watermarks on things just to ensure that they stay free. A few more housekeeping things as well before we fully dive into metabolism, in the introduction of it at least, is that my resources are entirely free. I have a work schedule that I take off time just to make free resources. So if there's anything that you want me to make, if you do like my teaching style, the way that my slides work, go ahead and request it in the comments. There's also a page on my website that you can request things, and I'll add it to my list of things to eventually work on. I can't make any guarantees, but I do have time to work on anything that you guys want me to. So it's one of those things that I'll just crowdsource whatever the most wanted topic is, and I'll work on that next. The last thing that I want to mention on this slide is that I am, of course, not perfect. I do a pretty good job of proofreading my entire script and all of my notes on the slides, but I do sometimes make quick little typos and things of that sort. So if I have made any corrections, I'll either mention them in the video later on or in the description, just in case anything pops up. So just to start introducing metabolism, basically anytime you eat food, that food is first digested, right? Digestion is that process of turning this chicken sandwich, which I'm just now realizing how hungry I am. It's this process of turning this chicken sandwich into the basic monomers that make it up. The basic glucose, basic fats, basic protein, amino acids that make up all of the ingredients that is in this sandwich. That is digestion. After digestion, your food has been turned into all of these individual molecules. Like I said, for our purposes, we're mostly focused on glucose for right now, but you will also have fatty acids and amino acids. Metabolism, on the other hand, is the study of what we actually do with this glucose. After digestion, that glucose is going to go into your blood and some of it's going to go to your liver. Metabolism is the study of what's coming after that point. So just as a quick reminder, you just want to make sure that you're not confusing digestion with metabolism. They're two separate processes that are interlinked with each other, but digestion is what's going on in your stomach. Metabolism is what's going on in your blood and in your actual cells. So just as a basic introduction for what you need to know for all of these pathways, you need to know each of the metabolic pathways that we're going to talk about. You need to know what hormone or metabolic state that promotes that pathway. You need to know the important enzymes of each pathway. We're going to talk about each one. You need to know the molecules that are used in that pathway, whether it's something like glucose or maybe a pathway uses ATP. You need to know if that pathway has used or produced ATP. You also need to know how much of it for several of the pathways, specifically how much per glucose, something like that for an example. Of course, we'll talk about the specifics when we get to every single pathway. You also need to know the location of every pathway, not only within each cell, but also which body part is doing it as well. And then finally, on my slides, I'm always going to bold and underline the most important information. So the easiest way that I recommend learning MCAT metabolism is by starting with the 3 metabolic states. These metabolic states are basically just the idea of, do we have energy in the blood or not? So there's 3 of them. The first one that we're going to be talking about, especially at the start of the video, is called the postprandial state. It's also called the fed state because this is basically the idea that you've just eaten a meal, and so all of your glucose and fats and everything that you're using as energy have come from your diet. You haven't used any of your long-term storage for anything. Everything's coming from the diet. That is the postprandial state. So insulin is going to be the hormone that's responsible for getting us through the postprandial state. It's going to promote all of the postprandial metabolic processes. Eventually, after a few hours, we're going to enter what we call short-term fasting, or the post-absorptive state, meaning right after the absorptive state, right after postprandial. So this is the point where your body doesn't really have any of that glucose from that chicken sandwich anymore, and it's going to have to start making its own. That's why we call this the short-term fasting state. So this is when your body is mainly going to start doing 2 different things. It's going to start making new glucose in the liver in a process called gluconeogenesis to release to the blood to get sent around the body. It is also going to be where we do glycogenolysis, which is this process that we are lysing or breaking down glycogen in order to provide glucose for our blood so that our tissues can use it. If fasting lasts longer than that, usually roughly around 24 hours, this is prolonged fasting or long-term fasting. This is a pretty desperate form of the fasted state that arises if you haven't eaten a meal in the past 24 hours. So this is basically what's happening when your body is starting to turn on its emergency reserves. The main processes that we're going to see here are fatty acid breakdown. It's also called beta oxidation, as well as protein catabolism, protein breakdown. So while insulin promotes the postprandial state, glucagon is the opposite. Glucagon is going to be catalyzing the increase of blood glucose. So it's going to cause all of these processes in the post-absorptive and prolonged fasting state that promote the production of more glucose, more energy into the blood. So I just find it very easy to organize the information for metabolism in my brain if I'm thinking about what is happening right after I've eaten a meal, because that's going to make the hormones make more sense. It's also going to make the exact processes make more sense as well. Before we begin, throughout all of metabolism, there's a few things that I want to make sure that we're all on the same page on. The first is electron carriers. So electron carriers are basically molecules that they're going to be receiving high energy electrons from molecules like glucose. And it's going to be giving these electrons to the electron transport chain, or ETC for short. By doing so, this electron transport chain is going to help us actually produce ATP. So these electron carriers, we say that they carry energy, but they themselves are not actually used as energy. What I mean by that is, for example, if you're trying to build a complex protein, you can't use an electron carrier to actually make that protein. You can only use the electron carrier to produce ATP, and then that ATP is going to be the exact energy that we're using to actually build those proteins. All of these electron carriers will be carrying 2 electrons at a time for our purposes. When they are holding their electrons, we say that they are reduced, and when they are not holding them, we say that they are oxidized. There are different electron carriers that are capable of donating 1 or 2 electrons at a time. You don't need to worry about that for the purposes of the MCAT. When the electron carriers are oxidized, meaning they are missing their electrons, they're waiting for these high energy electrons to show up, they are always going to have this plus at the end of their name. NAD+, FAD+, NADP+. That is how we know that they are oxidized. They're waiting for electrons, waiting for energy. When they are reduced, meaning they have now received those high energy electrons, notice that we have given them all a hydrogen. This is specifically a hydride, meaning that it's a hydrogen that is carrying electrons with it. So that's where we get the names NADH, FADH2, and NADPH. I'll make a note about NADPH here in a second. It's a little bit weird, but these are the forms of the electron carriers that we're trying to create by using glucose and these other metabolites. We're using glucose, the high energy electrons that glucose can carry, to give to NADH and FADH2 so that these electron carriers can go on and help us make ATP. To begin, we're going to start off by talking about NADH and technically NADPH as well. We'll talk about NADPH specifically here in a second. But so NADH is made up of two nucleotides. It has a basis in the structure of nucleotides. A nucleotide, just as a reminder, is a phosphate, a ribose sugar, sometimes it's deoxyribose if we're talking about DNA, and a nitrogenous base as well. So you need to understand the basic structure of nucleotides, but you don't need to memorize the structure of NADH here on the right. Identification is going to be your best friend with structures on the MCAT, so I recommend being able to identify a nucleotide. You don't need to be able to draw NADH from scratch. NADH is going to be able to generate 2.5 ATP for every single molecule of NADH we produce. It's going to do this by donating its electrons to complex 1, and then those electrons are going to get passed on, allowing us to eventually produce ATP. We'll talk about that whenever we get to the electron transport chain and oxidative phosphorylation. Finally, just as a quick note about NADH specifically, NADPH is going to be slightly different than NADH. It has a very similar structure, but it is very different in terms of its function. We are only going to be using NADPH for fatty acid synthesis, as well as eliminating what we call ROS, reactive oxidative species. So NADPH isn't really ever used to generate ATP. It's only going to help us make fatty acids, as well as protect ourselves from these oxidative species that we're worried about. We'll talk about NADPH whenever we get to fatty acid synthesis and the pentose phosphate pathway. The other important electron carrier that we're going to talk about is FADH2. Sometimes I leave off the 2, that's just because I'm lazy, but just know if I say FADH, I'm talking about FADH2. FADH2 is a type of flavin, and I recommend understanding this 3-ring structure that flavins have. Flavins all are going to have a very similar 3-ring structure. You don't need to be able to draw FADH2. I just would be able to pick out a flavin and know that FADH2 is a flavin out of a lineup. FADH2 specifically is going to be generating 1.5 ATP per molecule, so it is less efficient in terms of energy production compared to NADH. And it is going to be giving its electrons to complex 2 at the electron transport chain instead of complex 1. So they both have different entry points into the electron transport chain. NADH is going to go to complex 1, FADH2 is going to go to complex 2. The other molecule that I want to talk about, of course, is going to be ATP. Here's the loose structure of ATP. Not loose structure, this is the exact structure of ATP. Again, you do not necessarily need to be able to draw ATP from scratch, but you should be able to identify the basic structure of a nucleic acid, identifying that this is a nucleic acid out of a lineup. So ATP, or adenosine triphosphate, 3 phosphates, it is a nucleic acid that is the universal molecule of energy for every single organism that exists. Every single organism, it is either going to make its own energy through something like photosynthesis or eat something like we do in order to produce ATP. So humans specifically, and mammals in general, we are using the energy from glucose, those bonds in glucose or fatty acids or even proteins sometimes in dire circumstances. We're going to be taking that energy and putting it into these bonds of the phosphates that are in ATP. These are very high energy bonds that once we've built them, we can slice one off at a time, and that is going to release a whole bunch of energy that our cells can use to, for example, build proteins or fight infections. Anything that our cells are doing, they're going to need ATP. Metabolism is the study of how we make and use it. Again, just as a reminder, you don't need to be able to draw the exact structure of a nucleic acid. You just need to be able to draw the basic idea of what a nucleic acid means. So just to exemplify what I'm trying to say, you should know that on the 1' end, this end over here, on the ribose sugar, we're going to have a nitrogenous base. So a nucleic acid is going to have a nitrogenous base right here. It is going to be attached through the 1' carbon, that's this carbon right here, to a ribose sugar. If it's DNA, this is deoxyribose, but that's a topic for a different day. And then this ribose sugar at the 5' carbon right here is going to be attached to 1 to 3 phosphates. So that's what I'm trying to say when I say you need to be able to draw the basic structure of a nucleic acid. You don't need to be able to draw the perfect structure of adenine. You should just know that the base is going to be attached to the 1' carbon right here. And phosphates are going to be attached to the 5' carbon over here. In order to release energy, we're going to have enzymes throughout our cells that are going to be cleaving the bond between the gamma and beta phosphate. It's just a naming convention where the gamma is the 3rd phosphate away from the ribose sugar. The beta is the 2nd phosphate away from the ribose sugar. So basically, we are building up all of this energy in this bond right here between gamma and beta, and so that once we need it to be used for the cell, we just cleave that bond, that energy is released, and therefore we can do something like build a protein. Once that phosphate has been cleaved, we've gotten rid of that gamma phosphate, we now call the molecule adenosine diphosphate. The naming is the exact same, just notice that we have lost one phosphate, which is why we went from a triphosphate to a diphosphate, or in short, ATP to ADP. Just to finish out the naming convention, the phosphate that is closest to the ribose sugar is the alpha phosphate. Some other important things that I want to mention before we fully dive into the rest of metabolism is that these slides, and for the most part, whenever someone talks about metabolism on the MCAT, we are mainly referring to eukaryotic metabolism. Prokaryotes do not have membrane-bound organelles, so their processes are going to be slightly different. The steps are mostly the exact same, it's just because they don't have organelles, they get to skip a few things. And so I want you to assume that we are specifically talking about eukaryotes unless I specify otherwise. There will be a few places where we compare and contrast all of these steps in different organisms. Additionally, for all of these processes, you mainly just need to know the enzymes that catalyze the irreversible reactions. So if you see an enzyme on any of my slides, you can assume it's irreversible and highly regulated, unless you are told otherwise. Additionally, these enzymes are often going to be inhibited by a certain hormone, whether it's directly or indirectly, doesn't really matter. There's going to be a hormone that's going to be inhibited or promoting them. And there's also going to be a product that these reactions make, and that product is going to go back and inhibit its own production. If we have plenty of ATP, we don't need to make any more ATP, so ATP has a way of inhibiting its own production. This is called negative feedback, so those are the two main ways that all of our metabolic processes inhibit themselves, through a hormone as well as negative feedback loops. There's also going to be a summary at the end of each metabolic process and physiological state. This is also, of course, going to be listed in the supporting notes as well, just to help reiterate all of the most important stuff. There's going to be a whole bunch of information coming at you. I want to just reiterate that there will be a chance for you to kind of take a deep breath and make sure you understand everything I've told you with that summary at the end of each process. The processes on my slides that are promoted by insulin, whether it's directly or indirectly, are always going to be colored in blue. So if you see the color blue and I have a little key on the slide, you should just assume that that is an enzyme or process that's promoted by insulin. If it is something that's promoted by glucagon, it is going to be in orange. If it is a step that is producing ATP, then that is going to be in green. If it's a step that is using ATP, it's going to be in red. And then finally, if we are producing one of our two important metabolic electron carriers, those will be in purple. So that's everything I wanted to cover just as an introduction, not only to metabolism, but also the way that I do things over here. Like I mentioned earlier, check out the description of this video or the bio of my video, whatever you call it. That's where I'm going to have the notes listed. That's also where I'm going to have a link to my website listed in case you do want to check out my other free resources, my tips, or access my tutoring page, or request a new resource that I'll make for free at some point. That's also where you will find the notes that support this video, as well as any corrections that I may make to these slides along the way. Like I mentioned earlier, I've done a good job proofreading those, but I will read through the comments just to make sure no one has any questions or complaints.